John Bird coming to you with Liberty Now on air. And this is going to be a weekly broadcast. I'm going to be doing some interesting interviews and discussions about all things globalism, geopolitics, the New World Order, and the news behind the news.
Oh, yeah.
And I'm open to all perspectives, so I'm not going to try to tell anybody what to think. I just want to present the truth as best I possibly can. So I want to just interview people with their perspective on different things, and especially people who in my opinion still have some degree of common sense, who are willing to look at evidence.
Yes.
And at least consider all sides without getting overly emotional in an increasingly polarized world. I think we need more rational discussion. Anyway, we're chatting with Javier, Dr. Figueroa, Dr. X. This is my favorite name for you. That's awesome.
It's good to see you.
Yeah. So, Javier, you're in Seattle, Washington, in the U.S.
That is correct. In the great state of Washington.
Yeah, and where Katie and I had been living for the last 15 years before we moved here to New Zealand on the other side of the planet. And yeah, it's interesting to see how different things are between countries, but I think we're all facing the aftermath, or it's still ongoing, the outcome of COVID-19 in a COVID world in the last year. A lot of different opinions on what it's all about. There's a ton of stuff I want to talk to you about, Javier. Like I was saying before, we'll probably do a few different podcasts, maybe a few different episodes, because I've got only a 25-minute slot here, so I'm going to have to edit down to what I can for each one. But I thought a good starting point would be, the whole COVID thing. That's top of mind for a lot of people, pretty much everybody. And
Yep.
yeah, so I guess just introducing you, Javier, tell me a little bit about, what's your sort of CV, your background, your bona fide.
All right. Well, I'm originally an immigrant to the United States. 1973, my parents left Chile under duress after a coup that occurred coincidentally, 9/11, 1973.
Oh,
And basically, we emigrated and lived between the United States and Mexico for about 7 years until finally emigrating and settling down in California in 1980. And so I've been in the United States living, being educated since 1980 in California, in Washington, in Oregon, but basically in the U.S. So my first foray into the science and applied sciences was at UC Davis in the 90s. Received my bachelor's degree in microbiology from UC Davis, go Aggies. And
yeah.
and one of the best things about UC Davis is that they had the medical school there, and they had some excellent infectious disease researchers, teachers, virologists. So I got a really, I would say, a world-class education in immunology, virology, and microbiology. And then I did some master's work at CSU Fresno, where I completed the coursework for a master's degree in biotechnology. So basically applying the techniques and know-how of basically you need to grow antibodies, you need to grow cells, you need to grow proteins. That's what
Yep.
the degree taught me in essence. And that also, I didn't get my degree because it would have required another year of doing a master's dissertation or thesis. And that's when I was accepted for my PhD program up here in University of Washington, where I completed a PhD in neurobiology and behavior from the Department of Toxicology and Environmental Health. So I also have a background in toxicology from the UW. After graduating, I did 2 postdocs in the Department of Bioengineering, where I was exposed to engineers and the rigor that they have for doing applied medical research, as well as absolutely destroying a lot of the assumptions I held as a biologist and molecular biologist. The rigor was much
Yeah.
greater in terms of doing that. And then I did a second postdoc with Gerald Pollack in his lab, looking at water and water structure and water in biological systems, which is significantly different from water in the water that we drink. So that was
Right.
also another the great rabbit hole in my
Yeah.
education.
Some
That opened
really,
up a lot of stuff there.
yeah. I remember having some discussions with you about the implications of that, and it is some really interesting stuff. And so I guess it goes without saying that your opinion is not uninformed when it comes to this whole new COVID
Correct.
19 pandemic. I say that a little bit skeptically, but I've seen a lot of things that justify skepticism.
Yes.
And it's interesting what you can sort of discern from watching the mainstream media. They all seem to be pretty much lockstep and uniform in their opinion. It doesn't seem to be journalism. It seems to be more a narrative, very unified narrative. You don't hear much
That's
dissenting
right.
opinion. And I say that because I've seen, you know, doctors, and very well qualified people being censored or cut out of the
Oh, yes.
conversation. So that leads me to believe that there is something to at least question there. And then you have, off of the mainstream media, lots of things that are not allowed on social media or the primary alternate media channels. They're being censored left and right. But these are
Exactly.
not uninformed people, like yourself, Javier. So I guess, some of the basic questions. What is COVID-19? As
So
distinct from, say, other coronaviruses.
that's a good question. So SARS-CoV-2, which is the designation that they have for this virus, the official designation that they have for this virus, has genetic similarities to SARS, the sudden acquired respiratory syndrome.
Yep.
And what this particular virus has is an increased infectivity and what appears to be what they claim is a higher lethality. Well, I don't think they claim higher lethality, but it's certainly made to sound and look like it's more lethal than SARS. SARS had a greater lethality. It didn't infect as many people, but the people that it did infect, they did die from it. They didn't have that much resistance to it. What
Right.
we're seeing is that with SARS-CoV-2, even though there is sufficient genetic similarities to label it as a coronavirus, it has a lot more interesting features to it than the original SARS sequence.
Right. Yeah. So some of these additional features. Now, I'm hearing polar opposite opinions from supposed virologists who would know very well about this. But what I have heard from some virologists is that it made the jump from animals to humans naturally, organically.
Correct. Yeah.
And it was a strain. At first, it was claimed to be from bat or bat soup in a wet market, and then it was from pangolins or some other
Okay.
exotic
Yep.
animal. But these virologists I've listened to are making this opinion based on behavior and epidemiology. But the opposing opinions are from actual virologists and scientists who have examined it physically with electron microscopes.
Correct. Yeah.
So they are saying or claiming, I've heard that they can see that it's been spliced and it has insertions of other types of viruses, not the least of which is the HIV virus or a portion of it.
Correct. Yeah.
Am I saying that correctly?
So my understanding and my memory of what I was able to read from a study that was published by researchers in India that was then retracted
Right.
under pressure by the institute and under much criticism by I can't even determine if these were the people that were criticizing it were established virologists at either universities or institutional research groups. But they basically came to the conclusion that this was an engineered virus, that there were too many changes and too many changes that increased infectivity and potentially lethality for it to be a natural evolution of the SARS virus into this particular strain. There was another researcher, I think she was a Hong Kong researcher, that also came out and basically said, no, I worked with this, and she said that this was also an engineered virus. Now I don't know the veracity of the Hong Kong researcher's claims. She wrote a paper that went on the web as a preprint, and she made the claim that yes, this was an engineered virus. Now, as it turns out, the city of Wuhan in Hubei province is one of the only BSL-4 facilities for doing research on highly infectious and lethal viruses.
Right.
And it just so happens that was the city that got hit first with SARS-CoV-2, and
right.
all the connecting points and all the shifting narratives and stories that came out of China, and also the very strange behavior of allowing of stopping internal domestic flights or shutting down internal domestic flights into Wuhan, but allowing international flights
Right.
after discovery makes it highly suspect.
Right. I mean, at the very least, it's hard to say. Where's a smoking gun? Do I have a photograph of the genetic insertions? All I have to go by is the papers of the scientists who actually have done the research, and then we have inconsistent and suspect behavior, and then add to that the gain of function research being conducted by Dr. Fauci under the Obama administration in 2015. Am I correct?
Correct. So Obama in 2012 issued an executive order, out basically banning gain of function research in the United States. So Fauci, the NIAIDS director, basically signed off on funding research in Wuhan on gain of function of this, quote, bat virus.
Right. So we're talking about gain of function work from the U.S. This is presumably, well, they say for maybe the development of vaccines, but other people might say this is for military purposes or as a bioweapon. Either way, not safe. The United States says we need to stop this research. So for whatever reason, it was moved out of the country where they presumably have lower standards or safety requirements. And it's not only that, but it's coincidentally the same virus, right? They were doing gain of function research with COVID.
Correct. Well, I don't know if it's the exact same virus, but it's definitely in the same family.
Oh, okay.
And wouldn't you know it that it is derived from known bat coronaviruses. So there's enough sequence similarity to where you can say, yeah, this is a direct descendant or is highly connected to the bat virus research. And the principal investigator of the bat virus research, she worked at that facility.
Right.
So gain of function research is the claims that, well, we're using it to develop vaccines. I mean, yeah, that's nice. That's nice. What's the word I'm looking for? Um, that's a nice way to justify it,
Right.
but you can easily turn it into weapons. So this idea that you have to use weapons to make vaccines,
Right. Right.
give me a break.
Right. For the layman or people who aren't familiar with the term yet of gain of function, what does that mean?
So usually it means a change, either a natural mutation in the genetic sequence or a deliberate alteration of the genetic sequence, which either enhances the infectivity or the lethality of
Right.
a virus. Now, the problem is that you have to have a sort of an in-between point. You can have highly lethal viruses that are not going to be very infectious because they kill their hosts. So it's going to
Right.
be self-limiting in
Right.
some senses. But you can be a highly infectious virus that has very low lethality, which allows the virus to replicate and spread without killing the host. So you have that sort of in-between point. So a lot of the flu viruses are highly infectious but have low lethality.
Right.
You have some viruses, some severe influenza viruses that, for example, the 1918 influenza,
Right.
that is highly infectious and also highly lethal, but it burns out quickly.
Right.
And because of the changing nature of travel, you could get this widespread um, uh, uh, distribution.
Faster
Now, what's
infection rate.
interesting is that, yeah, you had a lot of deaths, but there's a lot of confounding factors that need to be addressed with the Spanish flu as well. But we're nowhere near the mortality levels when you adjust for a population.
Right. So yeah, when it comes to mortality rates, there's a few things that that need to be discussed that aren't
Yes.
allowed to be discussed on social media. One of those factors is hospitals being designated as COVID facilities have required big sections of the hospital be vacated, which has meant
Yep.
empty hospitals.
Correct.
Well, in anticipation of lots of, the 2 million dead that we were projected to have in the U.S., I
Yep.
guess. And I've seen lots of video showing empty hospitals, which would lead me to believe. And also, there's been faked video or staged video where the media has played it up. They've actually staged people in biohazard suits and gotten people to stand in line outside of the hospital to make it look as though people were waiting to get tested and get into the hospital. And it
Correct.
turned out not to be the actual case. And if it was truly as infectious as we're led to believe, that wouldn't be necessary.
Yeah.
So there's that. And then there's also the fact that hospitals in the U.S. were given a emergency funds to deal with the pandemic, right? And I believe that in the bill it was written that they would receive like $13,000 per COVID patient, and then another 50,000 or 30,000 if they were required to be on a ventilator.
Correct. Yeah. So
So
there was definitely some monetary incentives.
it's exactly that's what I wanted to get to is this automatically gives an incentive, right? Because the hospitals are no longer able to or restricted from receiving their regular flow of income from their regular patients, even cancer patients, right? Because the facilities have been redesignated for COVID. So
Correct.
so out of desperation for money, like any business, all the designated non-essential businesses, there is an incentive. Now you would want to say, well, but doctors have sworn an oath, to do no harm, and they would never do anything like that. But I mean, you have to at least look at, like, okay, there's monetary incentive there, and then
Yes.
and then you have anecdotally, cases. how widespread it is. It's hard to say because it's hard to get the global figures, but we know for sure cases where young people that were in a motorcycle accident came in, and they died from the trauma of the motorcycle accident, but they also did what's the test, the COVID test that they use, the PCR test.
Yeah.
And so if they tested positive, then they're counted in the COVID deaths column. So you have to question
Correct.
the accuracy to the number of deaths attributed to COVID. At the very least, you got to question some of those numbers.
So for the U.S., what's interesting is that there was a lot of changes associated with how people reported the death statistics in the United States. So the CDC in 2003 put out 2 handbooks for coroners and pathologists and medical doctors on how to properly categorize how people died. So since 2003, you've had nationwide a set of rules and criteria that told doctors how to classify. Someone had heart disease, what was the underlying cause of their death? So
Right.
if someone comes into the hospital and they have, they're diagnosed with the flu and they develop a pneumonia, and then, they're at the hospital for several days, they get a pneumonia, which usually can either be a viral or bacterial infection.
Right.
And all of a sudden they go downhill, nothing works, and they die. And they also had an underlying heart disease as well as diabetes. What was the cause?
Comorbidities.
The comorbidities. What was the cause of death? Well, if you have uncontrolled diabetes but stable heart disease, they'll designate that as, okay, well, the diabetes was probably the primary cause, with heart disease as a secondary cause that made the pneumonia brought on by the influenza the case. So the person died of diabetes or of heart disease or of these conditions.
So there's a standard hierarchy of reporting.
Correct.
Yep.
So what happened is that in March 24th of 2020, the CDC published the newsletter number 2 on COVID-19, which basically told them, we're going to designate a new way of categorizing deaths, and if someone has a diagnosis of COVID-19, that's going to be primary. So what I'm going to bring up, I'm just
That's
looking
interesting.
I'm looking for the actual paper. So there is this great paper done by IPAK, which is a group of researchers that looked into what happened with the way that doctors were coding deaths, especially COVID deaths. And this particular newsletter, this particular CDC change in the guidelines, is what actually turned the death statistics on their heads.
So I would love to get that link and add it to the podcast notes under sources.
You got it.
For listeners to go check out later. I encourage people not to just take at face value anything I or anybody else says. I really encourage people to question everything and to look at all the evidence before making up their mind.
There we go. So I'm going to do screen share right here.
Okay. Yep, I can see it now.
Okay.
Yeah, explain what we're looking at here, Javier.
So basically, the CDC, through various different groups, collects data nationally on all-cause mortality. And so what we have right here is basically looking at the week-to-week variation in all causes of death throughout the states. And so what I've done here is basically adjusted the year-to-year change in deaths by population per state over time. So what we have right here is deaths per 100,000 population for all causes in Alaska and Hawaii, for example.
Yep.
So you can see that right now, right? Is it
Yes.
visible enough?
Yep.
Okay. All right. So if we go down to, if we go, say, for example, to the West Coast states, which have a higher population number, you can actually see the seasonal variability over time. So you've got 2013, the tail end of 2013, 2014, 2015, 2016, 2017, 2018, 2019, and right now up to 2020. So around week 48 of 2020 is where the data is the most reliable.
Right.
So that's where it stopped. And so you see this, increase, decrease, increase, this wave-like pattern where you all of a sudden get some spikes.
Yeah. Would those be like
Those-
seasonal flus or?
Seasonal flus, yeah. And again, this is all-cause mortality, but you do get increases that exacerbate other conditions,
Right.
like heart disease and
Right.
things like that. So you get those spikes in there. So what's interesting is that California, Oregon, and Washington, you don't get we haven't seen those sudden huge spikes.
Well, and they all mirror each other too. They seem to follow the same pattern.
Exactly. So what happens is that we're seeing that instead of having these huge spikes, we're actually getting some extensions of slightly higher seasonal mortality, but not huge,
Right.
right? And then you start moving to states in the western states, northwestern states. You start seeing the same thing. You start seeing an increase at the tail end of 2020, but the question remains, is that because of COVID, or is that because now people are experiencing undue stress brought on by lockdowns, loss of income, suicide, alcoholism, and drug abuse?
Yeah. There have been.
That's the other
Yep.
problem.
Yep.
So I'll just go through these quickly. Arizona, Colorado, Utah, New Mexico, you start seeing at the tail end, you see those spikes. Then at the tail end, you start seeing the increases in mortality. You have that rise and then that fall.
Yeah.
And as we start moving further down, you start seeing, fairly similar patterns where you don't see that dramatic a rise in deaths except at the tail end.
Right.
In the Midwest and Northeast East Lake states, same thing. You do get a spike. For example, Ohio had a significant spike. I'm sorry, Michigan had a significant spike which dropped off, and it cleared.
Yep.
That's even with lockdown. So
Yep.
people claim, oh, well, it could have been so much worse with lockdown. There is zero evidence that lockdowns had any effect on the trajectory of the virus. Zero.
Yeah. I haven't seen any either.
Yeah, and I'll just go through these real quickly. You got Texas, Oklahoma, and Kansas. And then you got Minnesota, Wisconsin, Illinois, and Iowa. And here comes the fun one. So hold on to your hat.
Okay. Wow.
This is what I call the DC power block. Pennsylvania, New Jersey, Massachusetts, Delaware, Rhode Island, New York City, and New York, Connecticut, DC, and Maryland. You get a whopping spike in
That's
the first.
huge.
That's huge. It's completely out of bounds for the rest of the country. You see a highly concentrated increase in mortality in this particular area that's centered around the DC corridor.
On the mainstream media, I know that they've reported like New York has a higher concentration, but weren't we to believe that we had exceedingly high deaths in the West Coast as well?
Nope. I mean, slightly higher than normal, but nothing that would make you go, "Oh my God, it's New York City." If you remove these 10 states, and I'll get to the bottom of this here. Let me move down here.
Okay.
So basically, like for example, the Appalachia states, Virginia, Tennessee, North Carolina, West Virginia, Kentucky, and South Carolina,
Yep.
you don't see that, right? And they're right in the East Coast corridor there. Florida had its spikes, but nothing like that particular area in the DC and the Northeast.
Yeah.
So this is more of a timeline that reveals what's been going on in the United States. So if you actually remove those 9 states in the United States, you would be, the alarmism that we had in this country would not be as prevalent. So if you can see this, we basically have all-cause mortality with all 50 states plus the District of Columbia, and then the orange line is all-cause of deaths. If you remove those 9 states, you can see that removing 9 states did not have much of an effect on year-to-year variation in mortality up
Right.
until you get to 2020.
Yeah.
If we hadn't had that spike, this probably would have gone unnoticed for the most part in the early phase.
Right. And it's quite comparable overall to previous years going back to 2013 there, right? Or 2014. And to be, completely objective, somebody might ask, well, the states, those 9 states you mentioned, have some of the highest concentrations in close quarters, close living quarters. So, that could lead to faster infection rates and higher death rates.
That is always the argument that is used, but they also have the highest concentration of hospitals. They also have the best infrastructure in terms of access to medical supplies.
Yep.
All this that we had, that hospitals are being overrun, this, that, and the other. They had to turn back ventilators
Right.
at one point because they didn't need them.
And also,
So
these are not these are not all of the densely populated cities in the
no,
country. This is just one particular area. So why wouldn't we see? In fact, if the genesis of the patient zero was in Washington State, you would think that it would be the West Coast most populated cities that would see,
and
particularly
that
like Seattle or
I can tell you that when the orders came down, we started getting this nonstop coverage. I went down to Harborview, which is our trauma level 1 facility that was handling car crashes and all that, but was also receiving a great deal of the ICU patients. I went down there at the height of the pandemic. There was no one in the waiting rooms. When I asked the people working that were doing the nose swabs, asking them, "Are you getting a rush of people?" They said, "No." "How is your ICU doing?" They said, "Well, we're doing fine." So it's been sporadic and it's been spotty, and it hasn't been, quote, overwhelming the system. The only reason that you have an overwhelmed system is that you cannot find enough staff to run the hospital.
right,
Because in a lockdown, if you don't have someone to take care of your kids, guess what?
right,
You're going to take care of the kids.
right, right. And particularly if you're overly concerned that this is a genuine pandemic and world population reducing event.
And yeah,
So I think it's fair to be a little skeptical
I agree.
when you compare this to the hysteria that we've seen that's been justifying these lockdowns. Thank you for that. That's really helpful. So I've been seeing like lots of concern, media concern about the sudden increase in cases. But I haven't seen a concomitant rise, is that the right word, in
Correct.
deaths.
Correct.
So if it's not as lethal, if there are a lot of asymptomatic people running around, that's just logically less concerning.
Correct. Yeah.
So with regard to testing even, I've heard some controversy about how accurate the PCR tests are. I don't even know if this is the gold standard or the only test that's being used to determine infection. But from what I've heard of the PCR testing, they have to amplify the test to get genetic material just to register. And
Correct.
they're also talking about it's registering any corona family viruses. So does that necessarily indicate COVID-19 even?
So yeah.
What do you know
the
about
thing
that?
is that it's basically depends on the manufacturer and what they're using to detect for the virus. So the PCR test is basically there's I'll just go into a really brief description. So the PCR amplifies small sections of either DNA or RNA, and the tests are designed to amplify specific sequences. So for example, there are some conserved sequences for a coronavirus that all coronaviruses share. So the PCR tests will look for that to basically say, are we actually looking at coronavirus? And then they'll have primers that look specifically for unique sequences in SARS-CoV-2 or in SARS or other coronaviruses. So there's different levels of verification. So you can actually look at multiple sections of the for using PCR. The problem with PCR is that people think that it's you have it or you don't have it. It's not as easy as that. PCR can actually amplify errors. It can actually find things that look close enough to it, depending on how you design the primers, that'll amplify garbage. So you have to be
Right.
careful.
So how
So
qualified is the average tester, person conducting the test? Are these, virologists or nurses or nurses aides or, who's conducting the
yeah,
tests?
usually it's going to be laboratory technicians that are trained in clinical laboratory practices. So
Okay.
these are fairly well-trained individuals
Okay.
for the most part. At the beginning, you probably had a lot of people that weren't as strictly trained, but you've probably got enough people running through the system now that have been trained on it where they're doing a better job of it.
Okay.
It's not so much the training. For the most part, I wouldn't trust a seasoned principal investigator to run a PCR machine. I would trust the lab scientists that are working on the bench day
Okay.
in and day out to do their job. But just to be fair, there are several ways of running it, and there's several different kits that you can buy to test it, and there's several different controls that you run to verify, am I doing it correctly or incorrectly. The problem with PCR is the number of cycles, that is, the heating and cooling cycles that allow for the DNA that's copied to basically come apart and allow an enzyme called Taq polymerase to actually make a copy
Okay.
of the DNA as it splits apart. So the heating and cooling cycle, what happens is that if you have DNA strands, you have a sense and an antisense. Well, it's too uncomfortable to do that. They'll align and anneal with their complementary sequence. So
Right.
the only way that Taq polymerase can actually make a copy is when the DNA strands are apart.
Okay.
And so Taq polymerase will go, oh, here's an open reading region that I can grab onto and make a copy.
Okay.
So that's why you have this heating cooling cycle that allows all the DNA snippets to come apart together again, and you can amplify that.
Right.
What's critical in all of this is that the number of cycles that you do, the number of heating and cooling cycles that you do, will determine whether or not you can get an actual a true positive versus a false positive.
Right.
So the number of cycles, usually the lower cycles that you do, the more likelihood it is that you're actually detecting something that's there. Now,
Right.
the problem with PCR is that it can't distinguish between a person that's infected with constantly, with a live viral infection versus a person that's actually managed to overcome the infection, and there's only just garbage, strands of coronavirus that's already been, clearing out of the system.
Right. So if they're testing COVID positive, it could only mean that they've been exposed to that specific virus or something very near it in the family of viruses.
Correct.
Right. And, to justify hospitalization or action or even counting them as an active case, they would need to be at least symptomatic, right?
Correct. Well, see, now that's interesting.
Or pre-symptomatic or...
Usually, if here in the United States, here's the interesting part. Here in the United States, in order to be counted as a COVID case, you just need to test positive, or you need to have enough of the symptoms to test positive.
Well,
So if you're a presumed COVID case, then you're a COVID case.
by that logic, I could have had any number of different kinds of flu in the past and have remnants of it that my immune system has already dealt with.
Correct.
And I don't actually have it, but there's it's there in the DNA is there.
Correct.
Doesn't mean I'm a current
You're a
live
carrier.
case.
And there's a major problem with this whole asymptomatic transmission story that's being told.
Yep.
One of the largest studies was done in Wuhan, and this was published relatively recently in Nature Medicine. And Nature is one of the preeminent scientific journals. And in a study of 3 million people, they found no evidence of asymptomatic transmission of SARS-CoV-2.
Okay, and in the
So-
country of origin, no less.
In the country of origin, they found no asymptomatic transmission. The
Okay.
studies looking at asymptomatic transmissions have several deficits in that they're looking at very small samples, and in some cases, they claim that, quote, asymptomatic transmission occurred because a patient did not have symptoms when they were actually taking medicine to suppress their symptoms.
Ah, yeah.
So that's where you start getting into some serious, you really need to start digging into the literature to try and understand, okay, how do they classify it? What do they say? What are they calling asymptomatic? There is no asymptomatic transmission is not the driver of infections.
Right.
Even Fauci said that.
Right.
Fauci stated it bluntly when he said asymptomatic transmission is not the driver of infections. And SARS-CoV-2 is not transmitted asymptomatically.
This is the
There might
opposite
be,
of what we're hearing on, all the media, though.
correct.
That's everything to worry about is,
Correct.
increase in cases, but doesn't even necessarily mean transmission, let alone morbidity.
Correct. Now, what's interesting is that the, one to get back to the end of the PCR thing.
Yeah.
Cycle threshold is critical because the more cycles you do, the more chance you have for a false positive.
Right. I've heard this. Yep.
In Portugal, very recently, two British nationals were being held in quarantine in Portugal because they tested positive for SARS-CoV-2. And these two British nationals basically sued Portugal for discrimination, but also because the diagnostic tests that held them in place they claimed were flawed. And so the courts in Portugal basically ruled in favor of the British nationals, saying that yes, you're being held illegally because the PCR tests are unreliable. So there was one paper that came out in Clinical Infectious Disease Research. Let me get the dates correctly here. So I've got it up right now.
Nice.
Yes, November 23rd, 2020. The authors were Rita Jafar, Sara Aherfi, Nathalie Wurtz, Cleo Grimaldia, Van Thuan Hong, Philippe Colson, Didier Raoult, and Bernard Lascola from Instituto Hospitalu, well, basically from Marseille, France, and also from the Thai Binh University of Medicine and Pharmacy in Vietnam, working together.
Yep.
That basically detailed the pitfalls of the PCR test. So what they showed was that with 25 cycles, they could actually culture virus in 70% of those positive patients. When they
Wow.
went up to 30 cycles, they could only culture virus in 20% of the sample population. So the more cycles you do, the more positives you get. So you increase your
Right.
population size.
Right.
When they got to 35 cycles, they could only culture virus in about 2 to 3%.
Wow.
So you go from high sensitivity and specificity at lower cycles that actually show
Right.
a positive response to all of a sudden everyone's positive, or you get a much larger percentage of people with turning positive. And currently, the cycle threshold that is recommended globally is 35 cycles.
That was my next question. I was going to ask if there was a standard that's being used, and this is being recommended from the CDC or by whom?
So the World Health Organization,
WHO.
CDC. So that's a problem, and there's also bigger problems because the group that designed it was a German group that originally did the PCR tests. Was just there's actually a lawsuit in Germany against them because they're using this test or variants of this test to diagnose patients when there's what is the evidence that you're diagnostic.
Right.
So that's a big problem as well.
Really interesting. Hey, I'm gonna just for safety, I'm gonna cut the recording here and then start a fresh one, just because inevitably it seems like I don't know that it's stopped at some point. Yeah.
Yeah.
And I'll probably be able to break this into 2 or 3 episodes. This is really good stuff, and I don't want to miss any of it. Thank you for doing your research.